Hey guys, so for the second screencast in the genetics series, I wanted to get further into doing actual crosses. Now, for this example, I'm not going to go into specific examples. I will have a pencast for you to go over actual examples and how you get to specific examples. Right now, I'm just going to give you a broad overview of how to do crosses, and we're going to use the monohybrid crossing scheme as our example. Okay, so this is something that Mendel did in his research. Again, Mendel was using pea plants, and one of the easier characters to follow is petal color. So again, we're dealing with that flower petal color gene. And here's what he did. So another vocab term here for Mendel is he used true breeding plants. Now, if you have a purple true breeder and you cross it to another purple true breeder, all you get is purple. That's the definition of a true breeder. Same thing with white. If you cross white to a white true bred plant, all you're going to get is white. So these lineages, all they have are the white genes, the white alleles, excuse me, on the flower petal color gene. Now, to make a long story short, the genotype for those purple true breeders is big P, big P. So it's homozygous dominant, and the genotype for the white true breeders is little p, meaning it's homozygous recessive. So this one has two dominant big P alleles. This one has two recessive big P alleles. So Mendel wanted to know what happened when he crossed those. And what happened refuted the idea that blending occurred genetically in all organisms. Instead, what he got was all purple flowers. So this white phenotype disappeared, and 100% of this next generation, the F1 generation, was all purple. So that's 100%. Another term for these plants are hybrids because they're hybrids from this plant and this plant. We'll see in a few minutes that the genotype from these plants was big P, little p, which is heterozygous. And another term for this is hybrid. So if we cross these two hybrids, thus dihybrid cross, because they're hybrids for one gene, basically you're looking at genotypes of this being crossed to each other. So they're hybrid for the flower petal color gene. We're going to follow that into the next generation, the F2 generation. And what he saw in the F2 generation was that 3/4 of the plants were phenotypically purple, and 1/4 of those plants were phenotypically white. Now I'm going to go ahead and actually do the Punnett squares and show you how he got it, but this is the foundation for this dihybrid cross. The hybrid part actually occurs here, where you get big P times big P little p. So basically you're crossing heterozygotes, or hybrids in this case. So again, 75% here purple, 25% here white, because that's 1 divided by 4. A quick reminder about genetics. Genetics is basically looking at probability. Okay, so going back to that dihybrid cross, in this instance right here, where you're looking at the F1 generation, you're looking at hybrids, so you're looking at big P times big P little p. This is essentially flipping a coin. So if you have an instance where you are crossing big P times big P little p, you have a 50-50 chance of contributing either allele 1 or allele 2. You also have a 50-50 chance of contributing allele 1 or allele 2 here. So it's 1/2. They each get half a chance probability, which is 50%. It's very much like tossing a coin. So coins you might as well consider as heterozygous, right? So there's a head on one side and a tails on the other. There's a head on one side of this penny and a tails on the other. So of the two possibilities, 50% possibility heads, 50% tails. Okay, so this question, this box here is asking you, what's the probability of tossing two coins in a row to get heads? Well, it's a quarter, right? what's your probability of tossing uh, uh, two tails together? Well, it's also a quarter. Now, what's your probability of tossing a heads and a tail? That's a quarter, and then that's a quarter. So half the time you'll actually do this, whereas 25% of the time you'll do this, and 25% of the time you'll do this. Okay, so it's really a probability instance. But biologically, let's consider the possibility of certain different diploids given certain alleles to the next generation. All right, so consider this cross here. We're going to put the male here and the female here. So I'm going to make this the male side and this the female side. So the genotype of the male is big P, little p. So I'm going to put the big P here and the little p here. Genotype for the female is also big P, little p. So I'll put the big P here, the little p here. What this is representing, guys, is that in this diploid female, so one chromosome here, the second chromosome here, for this diploid female, one chromosome has the big P allele, and one chromosome has the little p allele. That's represented here. So half of her eggs are going to get this big P, and then the other half of the eggs are going to get this little p. Similarly for the males, because he's also a diploid, one of his chromosomes will have a big P allele, and the other chromosome will have the little p allele, meaning half of his sperm will have, well, in this case, pollen, I guess, the sperm and the pollen. Half of them will have the big P allele inside, and half of them will have the little p allele inside. So what we're seeing inside these boxes, inside of 1, 2, 3, and 4, is the creation of a zygote. Remember, zygotes are fertilized eggs, and they're diploid, meaning they have two sets of chromosomes, one set from mom, one set from dad. So for this zygote, zygote number 1, I'm going to bring this P in from dad and this big P in from mom, so two big P's. That organism's going to get this genotype. Likewise, the mom's going to give a big P here, and the dad's going to give a little p here, so that's this organism's genotype. The dad will give a big P here, mom will give a little p here, that's this organism's genotype, and in this case both parents will give little p alleles. So that's what I want you to think about, guys. For this person, or this plant rather, it's a coin toss whether he gives this big P or whether he gives this little p. Similarly, for the female, it's a coin toss also as to whether or not she gives to her offspring this big P or this little p. And then you'll see different percentages of each of these happening, right? So these zygotes will be created at different frequencies. The genotype ratio is 1 to 2 to 1 because you see one of these, you see two of these heterozygotes, and then you only see one of these little p guy. So 1 to 1, 1 to 2 to 1 ratio. Now phenotypically though, the ratio is different because this big P has the same phenotype as this big P and this big P little p. They will all be purple. So you have 3 of them, and then this little p over here will only will create white until you only have 1 of them. And again, this is because the big P is dominant here. So in this heterozygote here, and this heterozygote here, you're still going to get a purple phenotype because the P is dominant to the recessive little p allele. Okay, well, I guess I went into a lot of this already, so we'll kind of cruise through this again. The genotype of this flower that produces these eggs is big P, little p. During segregation, during meiosis, chromosome that contains this allele will be separated from this allele at metaphase 1, right? So the 2 chromosomes that contain this, I'm drawing them duplicated here as sister chromatids. These two right here have big P. These two right here have little p. During metaphase 1, they are separated. So that's what this segregation is showing. You can either have a big P or a little p. Same thing with the sperm, big P or little p. Now, depending on how they come together, you'll get different genotypes and phenotypes, which is being shown here. Okay. Now, for a test cross. Now, a test cross is something that we didn't go over a whole lot in class. Some of you tried it. I'm going to give you a problem like this on the test because I think it's valid. I think it's a good experimental type question. And the thing is that for these purple plants, you know the phenotype. The phenotype is purple. Okay, but you can have two different genotypes to get a purple plant. You can have big P, or you can be heterozygous and have big P, little p. Now, in this test cross experiment, I'm telling you, you already know, you don't need me to tell you, I'm telling you it's purple, but the genotype is unknown. So what do you do when you know the phenotype, but the genotype is unknown? Here's what you do. You take that unknown genotype plant and you cross it to a plant whose phenotype is known. You know that because it's staring you right in the face. It's white. And you know that the genotype. The only way that you can have a white flower is to be little p, little p. So you know the phenotype, you know the genotype, and then experimentally you set up the cross, okay? So you say, there's two possibilities. You can have a big P purple flower, or you can have a heterozygous big P, little p flower. Now, if I cross this purple P plant to a recessive, homozygous recessive white plant, so this is the white one over here, what's going to happen? Well, if it's big P, you are going to get all purple plants because the big P's are going to dominate the little p's, and you're going to have basically all heterozygotes with the all purple phenotype. Okay, but if your purple pea plant of an unknown genotype is big P, little p, well, that's going to be a different story when you cross it to an all recessive white plant. Okay, because if that's the case, then you'll only get purple plants when you create heterozygotes, but when this little p and this little p meets with this little p and this little p, you're going to get homozygous recessive and thus white plants. So this is how you do a test cross. I'm telling you ahead of time, those of you that are watching the screencast, this is on your test. Take it to the bank. It's on your test. So for those of you that pay attention and watch this and practice this, easy points right there. We're going to practice specific examples of monohybrid crosses and just random crosses. So if you didn't get the foundations here, watch it again. If you want to practice other examples like the homework sheet that I gave you, I will go over those in class and on a pencast in the future, but you really should practice it on your own because I'm not going to be there to help you with the test. So you gotta know this stuff before the test. Okay, I hope that helps. Talk to you soon.